Right, the first thing I'd like to say is if anybody has a problem with me referring to any of their articles or literature, please tell me and I will remove it. But this I just took off the web. This is not for any profit. I'm just trying to expose the nature of my research. And this talks about membrane biophysics as viewed from experimental bilayer lipid membranes. Those are the fatty lipid membranes of the cells, the cell membranes. Now, before I talk too much, I just want to say this is about membrane electrochemistry. And that is the way these things work are electrically. That's how your whole body works. And it is a must. It says here, it can be said that an appreciation of the EDL, which is the electrical, what do you call it, electrical double layer, and that's what the cells are. So it can be said that an appreciation of the electrical double layer is a must in order to understand the properties of biomembranes. Those are living membranes of cells, both at as well as across their solution membrane interfaces. And what it is it's like a polar interface. There's a neutral area between two differently charged surfaces, and those are the surfaces of the membrane. And you could read this whole article. It's very interesting, and I'm going to go on much deeper. All right, I'm just going to basically read to you what it says about these electrical double layers, and those are the cell membranes. And basically what it's telling you is that once the charges are separated, you have a positive on the outside of the cell membrane, and you end up with a negative on the inside, and you'll have a neutral zone in between. So it says once the charges are separated, the electrical potential difference is set up across the interface, that the interface of the two substances, and the EDL is formed, electrical double layer. Now, the electrical double layer is defined as an interfacial region between two homogeneous phases in what the charge constitutes have been separated so that each side of the interface has a charge, hold on a second, is electrified, and the charges on two sides are equal in magnitude but opposite in sign. Thus, the interface between those two regions as a whole is electrically neutral, and that's what accounts for all this ionic activity. It's like that semi-permeable membrane. It creates its own sort of membrane in between the outside and the inside. I mean, that's why they call it a membrane, I guess, but that's exactly what it does. And they're highly reactive to electrical potentials, and that's what this thing is all about. Now this you should look into, electrolysis. Now normally this is used in plating metals and extracting ores and things, but there is another process that's in biology, and it's the same process. And electrolysis is called decompositional potential, the decompositional potential of the electricity between the dirt and the creature, which occurs naturally in the soils, and this was proven by the University of Alberta. There is in wet soils that percolate down, they create electrical currents. It's absolutely been proven by them. And this is what I suspect has caused these different ionic invasions. Now, they might be nucleophilic invasions, or they might just be simple ionic invasions or reactions, whatever you want to call them, adsorption, resorption, whatever it is. But the chemicals that are in the organic materials are being electrically altered, and they are being driven by the bipolar silicates primarily that are in the soils. Now, I'm sure that if it was in a blood solution, it would be that would be the primary material that would interact. So these tests are all being done right now. I'm working on them, and I'm even going to be working. I primarily work on creatures that were alive, walking things. But I'm also going to do this with trees and other types of vegetation to see what effect that has. I see what the petrified forest trees are, and I'm just going to see if I can sort of work in that area and see what happens. All right, all this deep into chemistry here, and that's the only way you can ever understand this stuff. Now, this is all about amino acids. The R group determines the amino acid. Look at the chemicals. Hydrogen, carbon and hydrogen, carbon, hydrogen, a little bit of oxygen, some more hydrogen, carbon, hydrogen, some sulfur, whatever it is, hydrogen, carbon, hydrogen, carbon, hydrogen, hydrogen, hydrogen. This is why you end up with fuel, hydrocarbons. And that's what these lipids are made out of, these cell membranes. So once the internal organic stuff gases off, this stuff here, creating your fluids and things, your natural gas and methane and petroleum and bitumens, and then you get down to the kerogen material, the leftover carbon bags. And that becomes coal, and eventually that becomes diamonds. It's the process. It is the process, and that is a natural process, and it's just natural. Now, for this same result to happen from something else, I need to know what that process is that beats this process. And for life of me, I don't think somebody's going to come up with that, but I'm listening. I'm listening with both ears.